overall structure and overall applications. Let's go ahead and start with the applications of nucleic acids. First off, there's two types of nucleic acids. There's DNA and there's RNA. DNA is the master blueprint. So for example, if I were to create a house, I would need a master blueprint that an architect has in creating that house if I want to reproduce that house. Instead of houses, we're going to go ahead and create proteins. So that's one direct application of DNA. And in order to create a protein, you need a master blueprint, in which case DNA is the master blueprint. RNA is the other type of nucleic acid. Now, just as you would never give a rugged foreman the master blueprint to build a house, why? He might spill coffee on it. The cell never gives the building factors within the cell the master blueprint, because it would probably destroy it after making the house. So what the cell does is it goes ahead and it makes RNA. So RNA is you can think of it as a crude perishable copy. So just as an architect would go ahead and make a photocopy of the original blueprint, the body does the same thing. RNA is the crude perishable copy, which then the protein making tools within the cell then make proteins based on. Consequently, DNA would last the lifetime of a cell, whereas RNA only lasts about an hour. So as soon as it's done making its desired protein, then eventually the RNA is broken down and recycled. So, two important processes. First one is DNA replication. DNA replication, as a cell splits, it needs to make an exact copy of itself. So that's DNA replication. You then have transcription and translation. Transcription is the process of rewriting the DNA, which is what transcribe means. Rewriting the DNA to make a crude copy, so it converts it from DNA to RNA. How come it's not an exact copy? Well, for one reason, we do want it to be perishable. We don't want a permanent one floating around making unwanted proteins. So we need a perishable. So imagine making a master copy but on recycled biodegradable paper, for example, that will eventually not hurt the environment. The cell does the same thing, creating DNA to RNA. But it still maintains all of the important genetic material in order to create the desired biochemical product, such as a protein. Now, the next one is translation. So once you go from transcription to, use transcription to create your RNA, you're now going to use that RNA to create, for example, amino acids found within a protein. So going from RNA to, and using that RNA as a map to synthesize a new protein, we call this translation, because we're translating from RNA to the amino acids within the protein. The other name for this is protein synthesis. Whereas in other science classes where you look at the big picture, we're looking at the atoms up of DNA and RNA, and then talk about its replication and transcription and translation properties. So, DNA and RNA are basically polymers. They're composed of repeating units called nucleotides. So here is a fragment of a, in this example, a RNA strand. I'll talk about the differences between RNA and DNA in a second. But basically, it's a polymer made up of repeating units, as you see here. There's three types. There's the phosphate, the sugar, and the amines, also known as bases. The phosphate and sugar provide the structural support. So they provide the backbone and the structural support for the DNA strand. The amines are the actual letters, if you will, of the genetic information. So we have 5 different letters in the genetic code. Just as in real life you have 26 letters, nature prefers 5 letters. Those 5 letters are then grouped into unique arrangements called words. These words, the scientific term for these words are codons. So here we see the letters grouped into words, but the fancy word for those are codons. And the codons, during translation, they code specifically for amino acids. So one codon would code for a specific amino acid, such as AUG would code for methionine. In general, a DNA strand would be broken off into what we call genes. So this strand would continue on where you'd have multiple words, aka codons, and the codons would code for multiple amino acids. And basically it's all grouped together in one gene. And one gene essentially codes for one polypeptide or protein. Now, DNA, RNA have a lot more application than just synthesizing proteins, but as far as this class goes, that's what we're going to be focusing on primarily. Here is another picture, again showing the general breakdown. Nucleic acid broken up into nucleotides, phosphate, sugar, amine, and then the breakdown. Again, the bottom ones are used in the structure, maintaining the structure of the DNA. They make up the backbone. Notice it forms a helical pattern, and then the amines or bases make up the important genetic information. These are the 5 amines you need to memorize these bases. Here's an example of DNA. Now, you will be responsible for, on the exam, creating single and double strands of DNA and RNA. So make sure you understand the pattern, make sure you have the important nucleotides memorized. I'll give you something like this right here, ACGT, and then you will be responsible for creating all of this. So let's go over some of the basics of creating DNA, and then I'll start talking about the difference between DNA and RNA. In this case, just as in proteins, you had the what we call the starting and ending side, the N-terminus, C-terminus. In DNA, we have something similar. We have the 5' end, and we have the 3' end. And that allows us to assign directions, saying 5' to 3' or 3' to 5'. Now, and usually we talk about 5' to 3' or 3' to 5' when we're talking about how the strands in a double-strand DNA are arranged, or when we're trying to tell you the direction that enzymes are latching on and what direction they're going. So it's important to be able to recognize the 5' in and the 3' in. If I give you a strand of DNA like this, we would say that this is going to be the 5' in, this is going to be the 3' in. Let's give you insight into how they came up with the 5, 4, 3, 2, 1 primes. If we take a look at each one of these sugars, we assume that the carbon to the right of the heterocyclic oxygen is going to be carbon 1. And then if we start counting across, 2, 3, 4, and 5. Let's go to this one over here. Carbon 1, 2, 3, 4, and carbon 5. Notice in this phosphate sugar backbone, we have on carbon 5 connected to a phosphate. We also have carbon 3 connected to a phosphate. We assume that if we were to sit there and cut a fragment of the DNA, on one side of the DNA, the very last phosphate would be connected to the 5'. We assume at the very end of the other one, the very last phosphate that would be connected would be to the 3'. Usually what we do is, you may see it differently. Sometimes they have a phosphate here, other times they just have a alcohol present. This is a inorganic esterification when they're connected. So in this example, they're showing there was no inorganic esterification. There was no esterification of the phosphates and alcohol forming a ester. What do you need to know? You need to recognize the 5' in and the 3' in, and then I could ask you to go ahead and identify the 5' in and the 3' in. Next thing you'll notice is the overall pattern. In this case, it's always a phosphate and a ribose, phosphate, ribose. Notice on the phosphate, for the inner nucleotides, it always ends with a negative charge. Yes, so think of this as an inorganic acid where it loses its hydrogen on the outside. Yes, so overall DNA is negatively charged. It's able to balance the charges within a cell because there are floating around ions such as positive sodium ions or potassium ions, and those help balance it out when it's liberated within the cell's solvent, such as the cytoplasm or the liquid within the nuclear membrane. If this were actually not being used and in its original chromosome form, this would actually be wound up around a specially positive charged protein called a histone. And histones are positively charged, so this is one way that it maintains being wound up around histones. We'll talk about those in a second. And I'm getting off on a tangent here. The next one is the nucleotides. Notice how they're attached. So if I were to go back a slide here, in this case, on the very bottom, the hydrogen is replaced and connected directly to the sugar. The hydrogen is replaced and connected directly to the sugar. So let's go ahead and move on. DNA is normally found double-stranded, and in this case, there are two strands which are complementary to each other. This is important for replication. So if you're going to make exact copies, it's always nice to have a carbon copy. So DNA always comes with its own carbon copy. The carbon copy, if you will, is anti-parallel. So if one end is 5' to 3', the other end is going to be the opposite direction, 5' to 3'. And it's wrapped together, forming a nice double helix, where the amines are facing the inside and the two strands are held together via hydrogen bonds. In this example right here, well, before I get there, I want to go ahead and go forward a bit. Really quick. Now, the helixes are wrapped around these positively charged proteins called histones. And again, the reason they're able to wrap around tightly is because of the negatively charged phosphate backbone. So opposite charges attract. I do want to point out that just as in proteins, you had primary, secondary structure, tertiary. There's also primary and secondary structure in DNA. I'm not going to be testing you on that. Just be familiar with it. And for example, when we get into secondary structure, we talk about how it's wrapped in histones, and then we can go on to a tertiary structure, if you will, where it wraps up into its tighter chromosomes. So that's a quick overview. Let's go ahead and go back. Now I'm going to go ahead and talk about the difference between DNA versus RNA. So DNA versus RNA, since you're going to be asked to write DNA or RNA strands. In this example right here, this is a DNA strand. Now, one big difference between DNA and RNA is on the ribose. There is no oxygen on the second prime carbon. So no oxygen. So that's where we get the term deoxy. Deoxy in deoxyribonucleic acid. Now, the other big difference with DNA is it is the only one that contains thymine. So, if we go ahead and we take a look at this molecule right here, this is only found in DNA. So those are the two big differences. So no oxygen on the second prime carbon and contains thymine. Let's go ahead and take a look at this thymine really quick. If we go back a couple slides, this is thymine only in DNA. This is uracil. Uracil is only found in RNA. Notice the big differences between the two molecules. The only big difference is this methyl group right here. So this is called the methyl cap. So that's the only big difference. So if I wanted to sit there and turn, if I want to sit there and convert it from DNA to RNA, then all I'd have to do is add in my alcohols. And I'd have to convert the thymine to a uracil. So again, all I'd have to do to convert from a thymine to a uracil is to scratch out that methyl group. So now it's a uracil. So now you're looking at, well, provided this is a single strand to begin with. So if I converted a single strand from DNA to RNA, making these changes right there, now you've converted it from DNA to RNA. Now, it turns out there are special enzymes which can break down nucleic acids. When you have these specific characteristics of RNA, where you have alcohols present and you have uracil present, then these can be broken down pretty easy. So they can be broken down after 1 hour. However, if I convert it back to DNA, it turns out that the thymine, also known as a methyl cap, it actually protects from being digested, broken down. The no oxygen also protects. So if the DNA is ever exposed to nucleic acid munching enzymes, those enzymes could never break down DNA. So that's one way that we ensure that the genetic blueprint is maintained. So we do want to break down RNA, for example. Why? Because there are some proteins which are needed more than others during the different lifetime of a cell. So we're constantly updating our RNA that we want to expose to the protein-making molecules. Consequently, you don't want the old copies running around, so those are digested pretty easily. These are not. So that's the big differences. DNA, it's hard to digest by enzymes by removing those specific characteristics. In RNA, it's very easy for it to digest after an hour. All right, let's move on. You'll be asked, I will ask you to sketch either a single strand of DNA, single strand of DNA, or a single strand of RNA. I will also ask you to sketch a crude double-stranded DNA. So here are nice pictures of DNA, and I'm sure you can turn it into RNA. The hard part is creating the double-stranded DNA. First off, if I give you the letters, so let's say if I give you A, G, C, and T, that would be one strand, and you'd have to go ahead and create the complementary strand. So in this case, the complementary strand would be T, C, G, A. So this would be the complementary. So you would have to know how to predict the complementary strand. And it's very straightforward predicting the complementary strand. A is always going to be hydrogen bonded to a T, and G will always be hydrogen bonded to a C. I will ask you to create something like this right here because it's really hard matching up each of the amines for proper hydrogen bonding. I'll just have you summarize the phosphates and the sugars as P's and S's like you see here. The hard part is I will ask you to go ahead and sketch the individual required amines. So for example, if I were to sit there and give you a double-stranded DNA, and the initial letters that I gave you was A and excuse me. I've already asked. Let me start over again. If I were to ask you to draw me a double-stranded DNA, giving the starting letters for one of the strands as A, T, and G, you would draw something like this, where you have your P, S, P, S, P, S for your phosphate and sugar backbone. You draw this really long covalent bond just to make it easier for you. You also label it 5' to 3'. The hard part then is to go ahead and predict what the other strand is going to look like. So this is the obvious part, phosphate, sugar, and then you're going to draw a strand from 5' to 3'. You will have to have memorized the bonding pattern. Again, adenine will always bond with thymine, and it will be a 2 hydrogen bonds. Guanine will always be bonding to cytosine. So let's go ahead and take a look at this picture right here. Here is adenine. Here is thymine. One pattern that you can see, and I'm just going to go ahead and use my yellow highlighter, is that the lone pair of the tertiary amine will always bond with the hydrogen in the secondary amine. Always, always, always. So nitrogen, nitrogen. So if we go through the other examples right here, you can see that. So here we have the, and again, it's part of the heterocyclic, it makes up the heterocyclic structure of the amine base. So here we have the nitrogen. Notice it's 1, 2, so it's a secondary amine. So the hydrogen will always be bonded to the lone pair, nitrogen, nitrogen. Take a look at this one. Same thing. Lone pair, hydrogen, nitrogen, nitrogen. Secondary, tertiary, 1, 2, 3. Yep. Lone pair, hydrogen. So that's one pattern. The other pattern is, if you notice, you have our traditional organic amine. You notice how they always use two words for the same thing. You have your amines referring to the amine bases, which is the entire molecule, or you have your amine functional group. So in this case, I'm looking at the organic term, primary amine. So here we have a NH2 coming off. So the hydrogen will always be bonded to the lone pair of the oxygen. Hydrogen lone pair of the oxygen. Guanine and cytosine, you will always, always see two of these. So again, you have to absolutely memorize these. Now the hard part is being able to rotate. So for example, this one right here, I had to go ahead and rotate. I had to be able to flip them around, since on this one we needed the thymine, and on this one we needed the adenine. So you'll have to not only memorize these, but you'll also have to be able to rotate them. These are exam 4 questions, which are weeks away, but these are something that you have to practice with. As far as quiz question goes, I'd ask for a single strand only.